introduce Professor Tim Kamp. He's graduated from the University of Notre Dame and then did his MD and Ph.D. at the University of Chicago before he did his residency at the Johns Hopkins University. Then he went to the University of Wisconsin in Madison, where he became an assistant professor, and he was a practicing cardiologist at the same time that he ran his lab. He has numerous awards, and I won't go through them all, but some of the more prominent ones are the Schuster Prize for outstanding cardiovascular research, top 10 advances in cardiovascular disease research in 2009 by the American Heart Association, and Best Manuscript Award in Circulation Research in 2009. He has over 50 publications and is considered a leader in cardiac stem cells. We're happy to have him here and hear what he likes to tell us.
Thanks so much for the kind invitation to be here, and it's great to see old friends and meet some new friends here as well. What I'd like to do today is share some of our work on caveoli and how they may be involved in regulating cardiac repolarization. I did some consideration of the topic, so I hope you're not disappointed that it's not all stem cell focused. But I saw your previous speakers. I think you've gotten a pretty good dose of stem cells, so I'll try and diversify a little bit to share some of this work, although I will disclose that we do use stem cells for some of this work as well. So what I'd like to tell you about today is give you a little background so we all are on the same page regarding what cardiac caveoli are, talk about different subpopulations of calcium channels in heart cells, and how auxiliary subunits may be important in these channels, and also talk about a knockout mouse we've made, a conditional knockout for caveolin 3, the main protein isoform present in muscle cells for caveoli, and then talk about caveolin 3 and long QT syndrome. So we'll cover those areas today. I'll also disclose that I'm a founder for a company called Cellular Dynamics International, which uses stem cells for drug development and toxicity testing, but we really won't cover much of that today. So just so everyone knows what caveoli are and caveolins, the name comes from the cave-like structure that these proteins or these microdomains exhibit, and you can see that they are invaginations on the surface membrane. They're abundant in all sorts of different terminally differentiated cells, including myocytes, and they are partially defined by the structural proteins that are present there. Best known are the caveolins. There are 3 different genes encoding caveolins, 1, 2, and 3, and Cav1 and Cav2 are expressed fairly ubiquitously, but Cav3 seems to be largely specific to muscle cells, although it's expressed, we now know, in some neuronal tissues. And you can see here with antibody labeling for, let's see, I can use a pointer, I guess, for Cav3, that in this myocyte, you can see this is a myocyte from the sarcomeres, that Cav3 immunogold labeling is around these little cave-like structures. These bigger immunogold dots are CAV1.2, a calcium channel that we'll talk about later. But this shows there are many caveoli present in the endothelial cells lining the capillary that aren't labeled for Cav3. So that gives you an indication that caveolin 3 is a specific scaffolding protein present in ventricular myocytes and other muscle cells. And so we'll use that as a tool for understanding where channels are and how they can be regulated. The other bit of background I want to give you is so everyone is on the same page regarding the L-type calcium channel and its role in cardiac biology. It's important in maintaining the plateau phase of the action potential with the sustained inward current from the L-type calcium channel. L-type channels also play a role in pacemaker depolarization and conduction through the AV node. They're also known to be important in excitation-contraction coupling. As many of you appreciate, the influx of calcium through the L-type channel can trigger intracellular stores released through the ryanodin receptor and calcium-induced calcium release to stimulate myofilament contraction, excitation secretion coupling in the atrial cells, L-type calcium channels are implicated, as they are in gene regulation, a variety of calcium-dependent signaling cascades, and they're a well-known target for a class of drugs called calcium channel blockers. This is a schematic of the L-type calcium channel shown here. It's a four-domain protein, a voltage-sensitive channel with the cytoplasmic beta subunit, an alpha-2 delta subunit, with the alpha-2 being primarily extracellular, and a gamma subunit. So a multi-subunit complex. And so we're going to think about L-type calcium channels and caveoli a little bit. And the reason that we're thinking about this is because in previous work that we've now published a number of years ago, we tried to understand where different ion channel proteins and regulatory proteins were localized with regard to caveoli and using caveolin 3 as a tool to look at that. And one of the things that I would point out is that if we immunoprecipitate with an antibody to caveolin 3, we pull down certain signaling molecules of the beta-adrenergic adenyl cyclase pathway, including beta-2 receptors, adenyl cyclase, pKa, G-alpha I, and PP2A. But importantly, also, we can pull down L-type calcium channels as well. So this just goes to show you that if we use neonatal rat myocytes and measure L-type calcium currents and plot the peak current as a function of time and then add a stimulus for the beta-2 adrenergic system, salbutamol and atenolol to block beta-1, then we see an increase in current. These are small cells with small currents. But if we add an siRNA to caveolin 3, we block that, we don't see that increase in response to beta-2 adrenergic stimulation, suggesting that beta-2 receptors and the L-type calcium channels may be regulated together in caveoli in cardiomyocytes. And the average data confirms that representative series of experiments. So one of the questions from that early work is we think there are multiple populations of calcium channels in cardiomyocytes, some present in caveoli, some present elsewhere in the cell, such as in the surface membrane or in the junctional complexes. So can we figure out what's the relative distribution of calcium channels in caveoli? And what we do here is an immunoprecipitation experiment where we use an antibody to the calcium channel, main pore-forming subunit in the heart, CAV 1.2, and we radio label the cells with tritiated PN200-110, which is a radioligand specific for L-type calcium channels. It's also amlodipine for the clinicians in the audience. And when we immunoprecipitate, we see that we can pull down most of the tritiated PN200-110 binding with this antibody to CAV 1.2, a little bit in the flow through there. So the antibody can recognize the protein that binds this radioligand, which we think is the L-type calcium channel. And so if we do the same experiment with the control IgG, we find that we don't bring down much of the tritiated PN200-110, and most of it stays in the flow through. So the next experiment then is not to use an antibody to the calcium channel itself, but to use an antibody to caveolin 3, the scaffolding protein present in caveoli, where we can pull down most of the immunoreactivity for Cav3, and we see little in the flow-through. And our IP now contains a little less than half of the tritiated PN200-110, whereas the flow-through contains a little bit more than half. So I think on average, about 41% of the channels we think are present, in, at least in the mouse system here. We've also done it for dog cells, are present in, associated with Cav3, so presumably in caveoli. So this is a way to help us discriminate the relative distribution of calcium channels in cardiomyocytes and help know the abundance of those different pools. So how do the channels get to the different heart cells, different parts of the cardiomyocytes? What is responsible for the localization? And so we hypothesized as I'm sure many of you would, that could be a role for subunits, and particularly we were interested in the beta subunits of the calcium channel. And so we suggested that specific beta subunits may target the calcium channel complex to caveoli, thus providing the unique signature of the caveolar calcium channels. So the CAV 1.2 or the alpha 1 pore-forming subunit with a specific beta subunit. Could that help us define what channels are present in caveoli, not only define and identify those channels, but also control their physiology to some extent and their functional properties? The calcium channel beta subunit has been relatively well studied. There's a crystal structure. Several groups have provided crystal structures for different beta subunits, and basically it falls into the Maguk family. It's got a guanylate kinase-like domain as well as an SH3 domain, and then this less structured hook domain in between. There are 4 beta subunit genes present in the human genome, mouse, and other mammalian genomes to a large extent, beta 1, 2, 3, and 4. And there, to make life interesting, there are multiple splice variants present for these different beta subunit genes. And this, that you can't see too well, but this dendrogram shows the different beta subunits that Jason Full in my lab, using just simple mRNA studies, found evidence for these transcripts. And with the available antibodies, many of these were identified as well. So we think there are at least 18 different mRNA isoforms for the beta subunit in the heart. We tried to confirm this with antibody staining for the beta subunits that we had available antibodies present, and furthermore to look and see which ones were associated with caveoli. So, for example, if we used an antibody to the beta 1A subunit that is present in the heart, we saw that if we immunoprecipitated with Cav3, we didn't pull it down. It all came through in the flow-through and the lysate. If we added beta 1B, we could see immunoreactivity in the flow-through and the lysate, but not with the anti-caveolin 3 immunoprecipitation. Or if we used an antibody to beta 2C, we could start to pull down some Cav3 here, suggesting there may be an association. And another less specific beta 2 antibody, recognizing the C terminus of the beta subunit, so it identifies multiple subunits or multiple isoforms of the beta 2 subunit. You can see that came down with the Cav3 immunoprecipitation. Beta 3 and beta 4 failed to associate it with Cav3. So we think that the beta 2 subunit uniquely associates with caveolin 3 and may be important in targeting these L-type calcium channels to caveoli. We did, and this is work by my colleague and former trainee, but now assistant, soon-to-be associate professor at UW, Ravi Bali-Japali, who is very good at electron microscopy and did immunogold double labeling and showed caveoli here with the small dots. And if you look and zoom up with an antibody to the beta 2C calcium channel subunit, you can see the larger gold particles in some of the caveoli, suggesting the presence of this subunit specifically in caveoli with this immunogold labeling. So you've seen an experiment like this before where we used an antibody or siRNA for the Cav3, but now we're going to use one to beta 2C in neonatal mouse ventricular myocytes and look at beta 2 adrenergic stimulation of the calcium current. And basically what you see under control conditions, if you use beta 2 specific stimulation with salbutamol and atenolol to block beta 1, you get an increase in current. So you can wash out if you add norepinephrine plus prazosin to get beta 1 specific stimulation, you get an increase. If you then treat another group of cells with this siRNA to knock down the expression of the beta 2C subunit, you block the effect of beta 2 adrenergic stimulation, but you can still see the beta 1 stimulation. So again, an indication that Beta 2 adrenergic regulation of L-type calcium currents, at least in this cell type, is dependent on the beta 2C isoform, and also we think dependent on caveolar localization. So we're trying to define the regulation and localization of these L-type calcium channels in cardiomyocytes. So here's an experiment that Ravi did to see if the beta 2 subunit, when expressed in a heterologous expression system, which is HEK293 cells, where you express the various isoforms, including the L-type calcium channel pore-forming subunit expressed to largest extent in the heart, CAV 1.2. We can express different beta subunits as well as CAV3. And basically, if we do that in HEK cells that really don't express any of these proteins to measurable amounts with immunoglots without overexpressing them with transfection, we see that if we then immunoprecipitate with the antibody to CAV 1.2 to see if the CAV 1.2 associates with the CAV3 and pulls it down, we immunoprecipitate here, we don't bring down any CAV3. With either the beta 1A co-expressed, the beta 1B co-expressed, but if we co-express the beta 2C and 4 isoforms, we can bring down CAV3, suggesting that this isoform, again, may uniquely be important in associating with CAV3. Beta 3, nothing, beta 4, nothing. So again, evidence for some specificity, with a different way of looking at the association of these proteins in heterologous expression systems. We next wanted to try and understand more of the structural areas responsible for this association, and this brought us back to think a little bit about the structure of beta 2 subunit. And you can see on this slide that there are multiple spliceoforms present that we've identified in the heart, beta 2A, that have different amino terminus, N1, N2, N4, and I won't go into the details, but you have different splicing of these alternative exons. There's some highly conserved regions here in the SH3, and then in the hook region, less conserved, and then here in the terminal regions, with the C terminus being most variable. Or I'm sorry, least variable, sorry. And you can see other isoforms here, beta 2B expressed in the heart, beta 2C, and beta 2D that's actually a truncated beta 2 subunit that has unique properties. But suffice it to say, this gives us the variety that we can test by just testing these natural isoforms to see if we can figure out what regions of the beta 2 subunit are important for interacting. Based on that work, we found that it was the beta 2Cs that were important for the interaction, but we hadn't tested all the others. So for beta 2A, N1, N2, N4, we did find some interaction. So we took another tack because most of the variability here was in the N terminus, and we didn't see clear differences there. So we did progressive deletions of the C terminus here. So beta 2C, full length, and then truncating at 553, 459, and 418. We would see progressively shorter beta subunits on our Western blot in agreement with the prediction, but they would all associate with CAV3 until we truncated to 418. So somewhere in this range between 418 and 459 of the C terminus of the beta subunit we thought was important for the interaction for beta 2C with CAV 1.2 and the CAV3, the caveolin isoform. Interestingly, all of these beta subunit combinations resulted in comparable ionic currents. So that's just a little bit of background on our work studying my favorite ion channel, the L-type calcium channel, trying to define where it lives in the cell. And this is just a little summary diagram, oversimplified undoubtedly, to tell you that we think there are multiple populations of L-type calcium channels in cardiomyocytes. We focus primarily in ventricular myocytes. The story undoubtedly is a little different in atria and other specialized cells, nodal cells. But in ventricular myocytes, we think there is clearly a subpopulation of L-type calcium channels present in caveoli that are regulated by beta-2 adrenergic receptor signaling, ACAPs, and PP2A is present, as well as PKA, all associated with CAV3 in this microdomain. And so we think this is a regulatory macromolecular complex that allows this channel to specifically contribute to calcium influx, which may be less important for EC coupling, but more important in regulating signaling pathways. In contrast, there's clearly another substantial fraction of L-type calcium channels that are present in junctional domains opposed to the calcium release channel in the sarcoplasmic reticulum, but not associated with CAV3, associated more with beta-1 adrenergic regulation and potentially distinct ACAPs that can contribute to their regulation. And ultimately, it seems likely that we'll find evidence for other populations of calcium channels as our tools continue to be refined. So the next story I'd like to tell you a little bit about is that our road of studying L-type calcium channels in the heart and trying to define subpopulations made us question what does caveolin 3 as a whole do to cardiac biology, and what we decided to do was construct a cardiac-specific knockout of CAV3 and see its impact in the heart. And part of the reason we did that is because not only is the L-type calcium channel suggested to be localized to caveoli, at least a fraction of those channels, but a number of other channels, such as a pacemaker channel, a sodium channel, KR 2.1, and the list goes on with varying degrees of evidence. So our hypothesis is that cardiac caveoli could provide integrated regulation of cardiac repolarization by controlling the density and functional properties of multiple different classes of ion channels. So sort of a big hypothesis, and you'll see why we came to that idea. So first, a little bit about the mouse. Basically what we did here is we created a knockout mouse by putting LOX-P sites flanking exon 2 of the CAV3 gene, and then we could cross out so we could get this floxed mouse. We got rid of the neomycin, and then we can cross it with a tamoxifen-regulated alpha MHC mouse, which will cut out exon 2. So we're left here with a heterozygote if we had only one allele targeted, or a homozygous knockout of the allele. So we can either knock out one copy or two copies of CAV3. If we add tamoxifen, so this is an alpha MHC regulated mutant receptor regulated transgene that drives CRE. This was obtained in collaboration with Jeff Molketin, and so we crossed that with our FloxCAV3 mouse, added tamoxifen in the chow for 7 days, and you can see that in the double allele floxed CAV3 that we get a decrease in CAV3 on Western blot compared to the wild type. If we give tamoxifen, there's really no change in the level of expression, and average data, again, for both alleles floxed goes down over time and reaches a plateau after 1 to 2 weeks, a low level. And I will tell you that part of this knockout, not getting to zero, is because CAV3 is expressed in other muscle cells in the heart, such as smooth muscle cells. And so we can't, with these whole heart lysates, we don't separate the cardiomyocytes and the muscle cells. So we don't think this necessarily shows a very incomplete knockout in the cardiomyocytes. If we look at this another way, again, we can normalize the Western blot signaling, and with knocking out one allele or both alleles, we get a progressive decrease. In this particular experiment, though, what's done differently is we isolated ventricular myocytes, so we can knock out most of the Cav3 in ventricular myocytes. And some of the evidence for that, again, electron micrographs from Ravi Balajapalli, which shows caveoli in the wild-type mice. Here you can see these little cave-like structures. And with the double knockout, you see mitochondria, myofilaments, but you lose the caveoli. And so not only is there loss of the protein, but previous studies showed that caveolins are essential for the formation of the structures, the caveoli. So if we knock out that protein, we can knock out the structure as well. So one of the ways we looked at the mice after we did this knockout is we did telemetry. So we implanted telemetry into the mice, allowed them to recover, and then knocked out the gene and studied the phenotype of the mouse. And what we saw, first of all, is this is a QTC in a wild type mouse before, I shouldn't say a wild type, but one of the floxed mice before adding tamoxifen with a QTC of around 48. And then after we knock out with tamoxifen inducing the pre-recombinase, knocking out Cav3, we see an increase in the QTC that's significant. It's most prominent early in the day, less prominent at 10 p.m. Again, some of you are aware that the QT interval can show some diurnal variation, and we don't see this in wild-type mice. So a hint that this is impacting cardiac repolarization because the QTC is prolonged after tamoxifen-induced knockout caveolin 3. So if we then go on and look at action potentials, Yoga Markandeya in the lab has done these electrophysiology studies, and not too subtle of an effect on the action potential. This is a wild-type ventricular mouse action potential. And this is from the Cav3 knockout. You can see the APD50 and APD90 are markedly prolonged in these isolated myocytes. And this was a fairly striking finding and quite reproducible. So not only is the QTC prolonged, but in isolated myocytes we see what you might expect, a prolongation of the action potential duration. So the obvious question is what ionic currents are changed to be responsible for this prolongation of the action potential. One other thing that we looked at is if we get rid of those little caves or caveoli in the cardiomyocytes, one of the things that comes along for free when we're doing the electrophysiology is measuring whole cell capacitance, and we see that that's decreased by about 15% with Cav3 knockout, suggesting, at least with the myocytes we sampled, that there is some decrease in overall surface membrane area. But if we then started looking at the individual ionic currents in the knockout mice, first we looked at peak inward current, sodium current here. So these are sodium currents, and we see that in the wild type and Cav3 knockout, they generally show a similar shape and morphology. And if you look at the average peak inward currents plotted as a function of test voltage, you can see the IVs overlap each other. So there's no clear change on peak inward sodium currents in the knockout mice. However, if we looked at the late sodium current in these ventricular myocytes isolated from the Cav3 mice, we find that there is a late current that we can find in wild type, shown here in black. But in the knockout, that late current is increased, and if you add ranolazine to try and pharmacologically isolate the late current, you can see you go up to the blue current here. So if we plot this out, the late current as the percent of late current over peak current, it's around the range of 0.1%, goes up to 0.8% or so in the knockout mouse, and ranolazine gets rid of most of that late current. So we think that knockout of CAV3 increases the late sodium current. And this is work from John Mikowski's lab, suggesting that one of the impacts that could lead to action potential prolongation is more late sodium channel current. However, when we looked at the next inward current we chose to look at, that's the L-type calcium current in these knockout mice, we found that there was a difference in peak inward current shown here, the voltage activated inward currents at 3 different test potentials. And if you plot the peak voltages, the peak currents as a function of voltage, you see it's decreased over the whole range by about 12%, I believe, in the knockout mice. So there's a decrease in overall peak L-type calcium current in the knockout mice. And if we look a little bit more at the voltage-dependent gating, we didn't see clear differences in activation or inactivation of the current with steady-state inactivation measurements. But if we plotted the currents and overlapped them, we could see that the L-type currents in the knockout mice decayed more slowly. And this is shown as the ratio of the current at 50 milliseconds relative to the peak is increased in the knockout at 100 milliseconds as well as 200 milliseconds. So there's a slower decay of the L-type calcium channel current in the knockout CAV3 mice, suggesting that knockout of CAV3 has a couple of effects on L-type calcium currents. It decreases the overall density, but the remaining channels show a slower inactivation or decay. But perhaps the largest effect is seen on ITO, and probably not surprising given what many experts in this room realize is that ITO is the main repolarizing current in the mouse ventricle, and these are just raw currents observed with this voltage protocol in wild type and knockout mice, and you can see that the peak ITO is significantly reduced to about a third of the level in the knockout mice. And if we break this down, and I don't have all the data to show you, but into ITO fast, ITO slow, and IK steady state, that all 3 components are decreased significantly in this model. So an impact on multiple potassium currents present and important for repolarization in the mouse heart. We also looked at another current that's thought to be important in Caveolae, and that's IK1, but we didn't really see an effect, this is from Lee Eckert's lab, on overall IK1 density in the knockout mice. So the next thing we wanted to do is, we like mice. Mice are useful, but we also like stem cell-derived cardiomyocytes. So we took some iPS-derived cardiomyocytes and treated them with a short hairpin RNA to CAV3. But the first thing we had to show was whether CAV3 was expressed in the iPS cell cardiomyocytes. And so you can see a band here where CAV3 immunoreactivity is. We also found some of our favorite beta subunits and a faint band for the L-type calcium channel. Although if we immunoprecipitated with an antibody to CAV3, get a lot more immunoreactivity for CAV3, beta 2 subunit, and we can more clearly see the CAV 1.2 channel. Likewise, if we immunoprecipitate for CAV 1.2, we can see all 3 of these proteins that we think are important in this macromolecular complex that we saw in the mouse cells as well. And we throw in a little human ventricle here too, that we see these different proteins present in the human ventricle. So for the experiment we did, we then took the siRNA control and didn't see any effect on CAV3, but in these iPS cell-derived cardiomyocytes, we could really get rid of the immunoreactivity or the protein level that is detectable of CAV3 without really impacting another caveolin isoform or actin immunoreactivity. So this tells us that caveolin 3 is expressed in iPS-derived cardiomyocytes, that our calcium channel subunits that we've been previously studying in the mouse and in the canine heart are also present in iPS-derived cardiomyocytes. And so what will be the impact of knocking down CAV3 and in this model system? And basically what we've seen is that knocking down CAV3 prolongs the action potential duration, and the average data are shown here for APD 50 and APD 90. There's a significant increase in both of these measures in these iPS-derived cardiomyocytes. So we think that there's some overlap in the biology. Both mouse and human cardiomyocytes show regulation of action potential duration by changing the level of CAV3. Obviously, the details of the ionic current may be different in the human cardiomyocytes, and we're working to sort that out over time. So with that background of the role of caveolin 3 in regulating repolarization in the mouse knockout model and human cardiomyocytes, I just wanted to touch on our understanding of CAV3 and the long QT syndrome. This is related to CAV3 being implicated in LQT9, the 9th form of long QT syndrome being identified. But it's also, I think, relevant to point out that in a variety of pathophysiologies, that CAV 1.2 channels are reduced in advanced disease when there are changes in caveoli. And so that may suggest a particular mechanism leading to changes in the density of calcium channel in heart failure. In atrial fibrillation, as many of you know, there's a prominent reduction in L-type calcium current that we think is related to changes in the structure of the cardiomyocytes and alterations in caveoli, although that's early work. And finally, I'll tell you about some of our data looking at CAV3 mutations, and this is stemming from a collaborative study from Vada et al. And done with many patients from Mike Ackerman's clinical program, studying inherited arrhythmias with John Mikulski. And basically, what we identified were several novel mutations in the CAV3 gene that segregated with long QT syndrome, primarily S141R and F97C. And in this schematic of the CAV3 gene, S141R is present more in the C terminus, F97C is present more in the intramembrane domain of CAV3. So, again, what is the role of CAV3 in controlling repolarization, and it's associated with these long QT mutations? The initial study by Vada et al., John Mikulski's lab, showed, as did our knockout mice, that mutations in CAV3 would increase the late sodium current, but we were interested in looking at L-type calcium currents given our data in the mouse model. And so basically what we did is express the calcium channel with the main pore-forming subunit, the auxiliary beta subunit, and 3 different types of CAV3, the wild type and these 2 mutations. And we did whole cell patch clamp studies. And what we initially found when we used barium as the charge carrier is we didn't see much of an effect. And nothing was statistically different. If anything, the S141R showed smaller currents on average, which seemed too consistent with the long QT phenotype. Didn't quite reach statistical significance, and the student in the lab who was studying this got discouraged and put it on the back shelf for quite a while. And we thought more about the sodium current and late current being important for these CAV3 mutations, the F97C and S141R. And then he began to try the same experiment, but instead of using barium as the charge carrier for the calcium current, he used the more physiologic calcium and began to see some interesting things. And that is that with the mutation S141R, that actually with calcium as the charge carrier, curiously the currents actually got statistically significantly larger than the wild type control, and F97C currents overlapped with regard to the current voltage relationships to a large extent. So now there was something that may be consistent with the long QT effect, more inward L-type calcium current, perhaps contributing to a longer plateau phase of the action potential. And you can see those larger currents here with the S141R. Also, interestingly, if you looked at current decay, that with barium and a longer pulse shown here, 400 milliseconds. So barium looks at primarily voltage-dependent inactivation of L-type calcium current. The currents overlap. If you look at multiple test potentials, there's really no difference between wild-type, the mutation F97C, and S141R. They all show comparable inactivation measurement here, measured as the amount of barium current remaining at 100 milliseconds. However, if calcium is the charge carrier, it gets interesting. One of the mutations clearly delays the inactivation of the calcium current, and that's the F97C mutation, and that's seen at multiple test potentials, whereas the mutation that caused the larger inward currents actually overlaps the wild type. So if you've been following, that suggests that we have two mutations in caveolin 3 that actually impact the calcium current in different ways. One increases the peak current, the other slows current decay, both of which potentially could contribute to action potential duration prolongation and long QT. So with that, I'll just summarize the things I've tried to share with you today. That is that there's a subpopulation of L-type calcium channels encoded by the CAV1.2 subunit, primarily in ventricular myocytes that are localized to caveoli. They're associated with the beta-2 subunit, which may provide some targeting there, and they're specifically regulated by beta-2 adrenergic signaling. So if you think beta-2, something's going to work regarding caveoli. Caveoli modulate multiple ionic currents in the heart, which can coordinately regulate cardiac repolarization, and we've shown that the knockout impacts multiple currents, the L-type calcium current, the late sodium current, ITO, and potentially more. As you know, there are many ionic currents that can be studied in the heart. We've also focused some on some long QT mutations in CAV3, F97C, and S141R. They impact calcium channels in different ways. They can increase the calcium current density for the S141R and slow current decay, respectively. Wow, a lot of misspellings in that book. And finally, caveoli and associated ion channel signaling pathways serve to integrate signaling and electrical activity, and we think may play an important role in a variety of cardiac diseases that we'll continue to explore. And I'll just finish by acknowledging that this was the work of many collaborators and students in my lab, including Jason Fold, Jay Best, and Yoga Markandaya in Ravi's lab, Craig January's lab, Saguna Bali helped, and in Lee Eckert's lab, Ravi helped as well. And with that, I'll thank you for your attention and happy to take questions. Yes.
Just a short question. In knockout mice, did they have PR prolongation?
You know, they don't have PR prolongation. Because we have the telemetry data, we've looked at all the electrocardiographic indices that we could get out from simple telemetry, and we don't see PR prolongation.
So do you think that the caveolae are not important in the aveno tissue?
Well, I think the question is more the relative abundance of L-type calcium channels in caveoli in AV nodal cells and how that may impact their conduction properties. But the decrease we saw in overall L-type calcium currents in the ventricular cells was fairly small, and whether that would be enough alone to decrease conduction velocity, I'm not sure. You might know better. We can collaborate and model it.
While you have that slide up, how do you square that result with the idea that you're increasing calcium current in some of your CAV3 long QTs? Is it a gain of function in CAV3? Something?
Yeah, so I think that's an interesting question. So that's how we're working to put the puzzle together, is that the knockout obviously is a loss of function and shows specific alterations in the current, with a decrease in the peak current, but a slower decay, which one of the CAV3 mutations does show a slower decay. So maybe there's some component of that loss of function. But the other mutation may interact with the channel in such a way that it actually produces a gain of function, as you suggest. But again, how CAV3 interacts to modulate the channel needs to be looked at. We think it's by the beta 7. Yes, Tom?
On the same topic then, since you've only reduced the total L-type current by 25% there, and yet there should be no caveolin there, that means that about 25%, because you had about a 50-50 distribution, so some of them must be now somewhere else. Where are they, and are they getting, do you have an enhanced beta 1 stimulation and no beta 2 stimulation in the CAV3 knockouts?
Qualitatively, the beta 1 hasn't, is still present. Beta 2 is reduced. There is a little beta 2 regulation, but it's significantly reduced compared to the wild type. You know, the challenge, as you well know, with all of these knockout models is how much compensation there is. So we did this conditional knockout in adult mice to try and minimize the changes and redistribution and alternate subunit expression, but that certainly is an important consideration. Yes, Katie?
Yeah. Do you see any evidence that CAV3 is expressed in other membrane structures like T-tubules? And did your T-tubular structure stay the same in your CAV3 knockout?
So is CAV3 expressed in T-tubules? That's an important question, since that's where most of DC coupling takes place in cardiomyocytes. And we don't see much in the way of structural caveoli in T-tubules on electron microscopy. There may be a few near the mouth of the T-tubules, but once you get deeper in the T-tubules, we don't see morphologic caveoli. We do see some CAV3 immunoreactivity, again, more at the mouth of the T-tubules than deeper in. And so we think that caveoli are relatively sparse in T-tubules relative to the surface membrane, as is the CAV3 protein. And the second part of your question was, I'm sorry?
Did you see any changes in the T-tubular structure in your
Right. So the T-tubules didn't remodel significantly, at least with our initial look. We didn't quantify it, but just doing the standard immunolabeling or diet and EPS, we don't see big changes in T-tubules.
So on that topic, before I keep telling medical students that beta-2 adrenergic receptor signaling enhances calcium currents, which leads to enhanced calcium release, what you're saying is that it's the beta-2 signaling is all in the caveoli, not in the T-tubules, so there should be no coupling effect to calcium release. Is that, can you cross that out of there?
So that's, I think that's something that hasn't been fully defined. My working interpretation would be that the beta-2 adrenergic receptors, via increasing calcium influx anywhere in the cells, potentially ultimately can lead to an increase in stores, and so over time can increase contractility. But it's a much slower and less robust effect than beta-1 adrenergic stimulation.
And you can see that at the individual beat level, that you cannot get calcium release from SR enhanced by beta stimulation.
You know, we haven't done the line scan control, but we'll look at that. So it's a working model. Catherine's next.
What happens to caveoli in heart failure? If the cells get bigger, does the membrane change? Are there actually more of the structures, or are they different in size? And how does that, so what happens to the calcium channel then? Because you said the current went up.
So heart failure, and maybe you and Igor know more since you've been working more with human heart failure tissue, but heart failure, there's conflicting data in the literature. Some data suggests CAV3 goes up and caveoli go up. Other data suggests that there's a decrease in caveolae and Cav3. In the dog tachycardia model that we studied, we saw a decrease in Cav3 and caveolae. Nowhere close to an absence, but a relative decrease. And how that impacts L-type calcium channels and doing the specific experiments to dissect out the different subpopulations we haven't done. So there are changes in L-type calcium currents in end-stage heart failure. The density decreases, which could argue is consistent with the loss of caveolae, but is that a one-to-one correlation? Don't know. And I don't know if you've looked at it with Ian.
Not yet,
Okay.
but now definitely will look. I actually wanted to follow up on Corin's question about beta 1 versus beta 2. So in your cartoon in the middle, conclusion, you placed beta receptors, L-type calcium channels, etc., but G proteins were missing. So can you?
I ran out of room.
So what is your take on place of different G protein isoforms in caveolae, and how do we couple to beta 2 receptors in particular? Yeah, here it is.
So, interestingly, we haven't gotten the specific reagents to define all the different G proteins that are known to be present in the heart, which is not trivial. Some interesting observations we've made is that GI is associated with Cav3. So if we immunoprecipitate Cav3, we bring down GI immunoreactivity, and that was shown in an earlier slide. But if we immunoprecipitate with CAV1.2, the calcium channel antibody, we don't bring down GI. So it suggests that somewhere in the caveolae, or to make life a little more complicated, there's some evidence that there may be distinct populations of caveolae containing distinct signaling complexes that may be more GI regulated complexes. GS comes down, alpha GS comes down immunoreactivity with both Cav3 and anti-CAV1.2 antibodies. So there is some evidence for differential G protein localization in caveolae and associated with the channel. But obviously there's whole projects that could be made to define different G proteins. So I apologize to the G protein biologists for not putting it on the cartoon.
And can I ask you a question? What is this PKC? It doesn't show what is the isoform.
Well, that is an area of debate. We think it's based on some of our experience. We think it's PKC epsilon from old experiments, but there's publications in the literature have suggested PKC alpha as well, and so there's some debate on that. It's probably species specific and the usual caveats. Yeah.
So one of your caveolin mutations actually specifically increase the current only in the presence of calcium. So you didn't show the calcium-dependent inactivation for that particular mutation. Did that have anything to do with calcium-dependent inactivation?
Actually, there was a quick flyby slide of that. Trying to fly by. Where we don't think, so you can't see it too well, but the blue,
Oh, okay, so there are
blue
two
is
traces.
overlaying the black here.
Yeah.
So blue and black, the blue is the mutation, the black is the wild type. So it didn't really clearly impact calcium-dependent inactivation.
So what do you think happened there? Why is calcium specific?
Yeah, I think that's a good question. Obviously, we think it's potentially an impact on either density or gating of the channels. It's probably density, since the gating seems to be fairly comparable for the inactivation amount. And so somehow it must be stabilizing more channels at the membrane is our working idea. In the presence of calcium, which is known to regulate multiple of the scaffolding proteins, as well as calmodulin on the channel itself, which is sort of another intrinsic subunit.
So why do you think that some of the channels are targeted to the caveolae rather than the sarcolemmal membrane? You made a comment about loading up calcium stores. Do you think that they're functionally separate and that's why they're targeted there?
So I think the caveoli are nodes for signaling. So there are a number of different signaling cascades present there. Not only beta-2 adrenergic receptors, but some of the tyrosine kinases and potentially other calcium-regulated pathways that may respond in ways, for example, to the activity of the heart cell. More action potentials, more calcium flux may integrate into those signaling pathways by the calcium localized caveoli, sort of the global model, and that it's more important in regulating signaling pathways and sort of an electrical signaling coupling is what we would like to imagine, although we have scarce little data to prove that. Yes?
Do you know if there's developmental regulation of when it's expressed? For example, is it higher expressed in the adult than newborn, and could that be responsible for some of the differences in the calcium currents of the lesion?
So the caveolin 3 is expressed higher in the adult heart than in the developing heart, and we see that recapitulated in the iPS cardiomyocyte model. So there's more CAV3 the longer they're maintained in culture. Whether that explains the differences in calcium currents, we haven't systematically looked at that. That's a little complicated because there can be differences in calcium channel isoforms expressed over time as well, both pore-forming and beta subunits. So I don't have a clear answer for that. Yes?
You just raised my question. The stem cells you used, can you tell me where they were from? Are they from your patients or cell dynamics or?
Actually, those experiments were with eye cells that are from cytogenetic.
And you just mentioned the maturation went well. How old were those stem cells when you looked at?
So generally they select those, so those are purified cardiomyocytes. They select them and cryopreserve them after about 25 days. And then we thaw them and culture them for about 10 days in culture before we study them. So depending on how you run your clock, they're 35 days roughly.
And my second question is it's just been interesting to me. Jonathan's patients had a long QT, is that correct?
Yes.
Okay. Did anyone try to see if calcium blocker shortened QT in those people?
Not to my knowledge.
I mean, that would, if I'm understanding properly, that's, to me, that's the question.
Yeah.
And the follow-up would be, is anyone taking stem cells from those patients to see if the calcium blockers work?
Well, that would be us, and we've been trying to do that for a couple of years. It turns out that a skin biopsy sounds benign to some of us in the room, but patients, when it comes to somebody taking a piece of skin, sometimes they can be a little skittish. So we don't have those yet, and these are not common mutations. So we're probably going to end up doing it with genomic editing to get those mutations.
Well, isn't CDI going to blood samples now? They're trying blood cells?
Yeah, so reprogramming is going more and more to blood. They have that technology. I don't in my lab yet. But yes, things are changing to blood. Okay, thanks so much.